Alright, welcome back guys. This is going to be one of our last two video lectures because our official end date is May 15th, this Friday. So we're going to wrap things up this week and we'll go from there. So there's no quizzes this week, there's no science dailies, there's no discussion boards, there's just an assignment over this phylogenetics set that we're going to be talking about. So if you have any questions on that, please email. I'm sure there's going to be a couple. I'm going to try to go over a little bit of that with the video today, so you have a brief idea of how to work that. But this is kind of a big thing that they talk about in college and classes like zoology and whatnot. It's starting to become more of an intricate part of those college studies. So I want you to have a taste of it and understand a little bit of it, so as we move forward, you know what to do. Okay? So another item on the topic is that CMU would like for you, if you are enrolled in dual credit, to fill out the course survey. You have to go to the CMU website, which I've posted in the Google Classroom, click on the My CMU, and then enter your student ID and password. If you don't have your student ID or password, or don't know them, or forgot them, I can email them to you. I have them, so all you gotta do is shoot me an email, and I'll send that out to you. It takes maybe 5 minutes at the most. Okay, it's just the feedback that they would like. They had very few people fill it out across the state of Missouri or wherever they offered this, so they were just wanting a few more by Friday. So if you could, help them out, fill out the survey, and they get a little feedback on how to improve the courses or whatnot, or your college experience. So thank you guys for taking the time to do that, and thank you for tuning in this last week. Alright, so we're going to go ahead and get started with Chapter 18, talk a little more about this. So we've been talking about morphological traits, and now we're going to talk about genes. So in the past, we looked at morphology traits, and we're trying to determine how species were related. Now we're going to get into the more modern approach, which is genetics. And there's a lot of interest in this field because it's starting to shed light on new paths of a lineage for how things evolved. Even as we look into like viruses, like the COVID-19, we see how some of these viruses have evolved by looking at their DNA and how closely related they are, which can also perhaps help us identify certain vaccines or treatments for these things. So it's kind of an important deal. So the kind and number of biochemical similarities among species are clues about evolutionary relationships. So molecular clocks are used to determine how far apart these organisms are, or how long ago they evolved. So a molecular clock is used to estimate how long ago two lineages diverged by comparing the DNA or protein sequences. Over time, neutral mutations that have no effect on survival or reproduction accumulate at a constant rate. So what that means is every organism, even if it was, say, the human species, will accumulate mutations over time. And what we're doing is we're accumulating mutations to essentially our non-coding DNA or their silent mutations. So even if you had a very important gene that coded for a very specific protein, say like hemoglobin, it's still a possibility you could have a mutation to it, but yet again, it still codes for the exact same protein. How is that possible? Remember, as we talked about the amino acids, there are 20 different amino acids, but there's 64 different codon combinations for those amino acids. So there are some of them that duplicate and have the exact same amino acids, but makes the exact same protein. So as we look at species, sometimes they accumulate these over time, and we can say, well, you know, humans and chimpanzees, yeah, they're close related, but there might be a key difference in a particular gene, indicating that these two populations have diverged a number of years ago. So the accumulation of neutral mutations in the DNA of a lineage acts as a molecular clock. The number of differences between genomes can be used to estimate the relative times of divergence. Now, that's where it gets to be kind of a gray area. It's like, how long does it take for a mutation to occur? Because they are random. So they kind of come up with this time scale, a clock of when a mutation could happen, how long it's been apart, you know, do you see it prevalent in many populations? So they have a number of factors, but are they 100% accurate? No. Most of the lineages that they give you, or divergence, it'll be a range. It could be, say, I've read some scientific articles on rattlesnakes, and they'll talk about anywhere from, say, you know, 500,000 years to 1,000,000 years. There's 500,000 years in there, of which they may be right or wrong. So again, it seems like, wow, that's a big time gap, but in the large scheme of things, that narrows it in within 500,000 years. DNA and protein sequence comparisons. Some essential genes are highly conserved, which means they are important. You gotta have these. If you don't, they mutate too much, you die. Okay, so the DNA sequences have changed very little over evolutionary time, and other genes are not conserved at all. So some genes can just mutate like crazy because they don't do anything. Comparing the nucleotide sequence of a gene or amino acid sequence of a protein can provide evidence of an evolutionary relationship, as we said. Generally, two species with many identical proteins are likely to be close relatives. So if you find two species, even though they may look physically, different, if you start looking at their biochemistry and their proteins, and they're exactly the same, that means there has been some physical changes, but not necessarily biochemical changes. And if you see some major biochemical changes within the organism, that means there had been some significant mutations and some significant time in between them, most likely. So generally, two species with identical ones, again, are close relatives, but if they're farther apart, a lot different. This is a picture from the book. This has those honey creepers that we talked about at the beginning of the chapter or the unit, and as you can see, there's a whole bunch of them there. And what they're looking at is the amino acid sequences, and they represent the amino acid sequences with these different letters. So the black ones represent ones that they all share. The red ones are ones that have changed. So as you can look in the first row from C down to M on the left side there, most of them, the first 5 or so, 6, they had the exact same amino acid, but the last 4 have something different. So they've changed a bit. Now, humans are in there, of course, so there's a couple other ones in there, but ultimately, you can see how they've changed over time ever so slightly. Now, you'll notice the dashes in there too. That means there was either an insertion or deletion over time. So there's either extra ones or some of them just got deleted out altogether. All right. Now, as we look at this one, we go down the line here, we're going to talk about some other ones, but it says DNA comparisons, DNA from nucleotide, mitochondria, and chloroplasts can be used. So absolutely, anything with DNA can show an evolutionary relationship over time. The mitochondria sometimes is used to show lineages from the maternal line, which are inherited as an intact from a single parent, usually the mother. Any differences in the mitochondrial DNA sequences between maternally related individuals are due to mutations, not genetic recombination or fertilization. So they're right in that aspect that there's the mitochondrial DNA doesn't typically interchange and cross over like you would see in us for producing gametes. So it's basically the same thing. However, we do know now, due to recent studies, that at least in humans, some of your mitochondrial DNA or mitochondria may have been inherited by your paternal line, which is your father or whatnot. So not every single case do we know is strictly maternal. That has really changed a lot of things in science, as we talked about before. But as you can see in this photo, you do have all of these different honey creepers now, and they have the DNA sequences. So as you look across, you'll notice the red ones. That indicates a mutation that has occurred. Now, many of these species are closely related, but as you notice, a few nucleotides have changed. Some of these may be silent or neutral mutations that didn't have any effect on them, which means the protein is still the same, but yet we can see there are some subtle differences between each one, which is kind of neat. And that's how scientists do this. And they can determine if there's more mutations, that means there's probably more time in between them, which means over time, there has been a whole bunch of things happen. So a greater distance between them, those two species. They can take that and use the molecular clock and build an algorithm to make a cladogram. And as you can see here, they've taken those species on the previous page here and put them into that cladogram. So now you can see how closely related they are based upon that particular gene or segment of DNA. And that's what you guys did with your cladograms using the GenBank website. You found some species or genes and you threw them on there and seen how closely they were related. And if you looked in the lower left-hand corner of your cladogram that you made on GenBank, you will notice there was a timeline, or should have been a timeline, of distance. So as we look at the branch of a cladogram, certain ones, it will tell you each little segment represents kind of like a distance on the map, how many thousands of years or hundreds of years that have accumulated since that mutation occurred, or distance between them. So take home message: mutations change the nucleotide sequence of a lineage DNA over time. Lineages that diverged long ago have more differences between their DNA and amino acid sequences than lineages that diverged recently. All right. So that is the end of that section, but I'm just going to talk a little bit about this and introduce you to your new assignment. So comparing patterns of development, and these are very important. So the pattern of how an organism develops with, from a zygote to an offspring is very important, because if you mess that sequence up, there are many things that could happen and usually would result in the death of the organism altogether. So having this plan or template that is highly conserved is important. So mutation in a master gene typically halts development, like I said, usually leads to death, but not always. Homeotic genes encode for transcription factors that determine details of a body from form during the embryonic development. So example, the floral identity gene affects the petal formation across many different lineages. It is likely that this gene evolved in a shared ancestor. So sometimes these are called Hox genes, which we're going to talk about. And within the segment, they code for certain body developmental plans. If you mess up this particular gene, it'll affect that particular area. So in fruit flies, we're going to talk about later how they mutated or duplicated this gene, and it started growing legs from its head, which is kind of crazy. But the fly did actually live, but now instead of antennas, it had legs. Okay, we're going to stop there, and now I'm going to talk about your assignment. So I found this assignment online a couple years ago, and I modified it. It's called the Beasties Phylogenetic Tree Activity. I turned it into the Beastie Boys because I like the Beastie Boys and whatnot. So while you're working on the assignment, you can check out who the Beastie Boys are. My favorite song is Sabotage. All right, so in this exercise, what you have here are a couple different little organisms, alien organisms. And if you notice, they all have these unique traits about them. They have little antennas, they have teeth, they have eyes, tails, legs, but not all of them have it. So the question is, how did these particular 7 organisms evolve over time? What traits emerged new, and which ones were always there is the question. So what we have here on the right side is called the outgroup, the base, the standard. So he was the first one essentially, and everything afterwards has evolved from this particular individual. All right. So what you're going to do with this assignment, and it outlines everything that you need to do here, is you have 2 tables. All right. So table 1, you're to find some characteristics about these little Beastie Boy organisms. Now what you're going to do is take these little guys and create a list of characters on the left side. And I've gotten most of them for you. Spots, teeth, eye size, antenna, feet. You have to come up with the final 3. Okay, so if you look at them, you'll find the other 3. Then you have to determine what is the standard. What was the base? What did the outgroup have at first? All right, so you have absent and you have present. So absent, you give a 0. We're going to use a binary code, 0s and 1s. So if we look at spots, we have the absent, which is going to be 0, and the present as a 1. So we go back up and we look at the outgroup. Does he have spots? No. So we want the outgroup to have all 0s if possible. Okay, but we see spots has evolved over time. So the outgroup would have a 0, as we know, and then those other 2 would have a 1 for having spots present. Teeth, you have flat versus sharp, so you have 2 different types now. So it's not absent just versus present, it's an actual type. So teeth, as we look up here, the outgroup has flat teeth, and again, flat teeth would represent 0. So then the sharp teeth, we see that 1, 2, 3, 4, 5 of them have actually evolved that. So they would get a 1 for that particular character trait. Then we go to eye size, and we look at eye size, and you can notice that some of the eyes are really tiny, but some of them are really large. So something new. So again, the outgroup, we'd want maybe small eyes, and then the evolved trait, large eyes. Okay, so you continue filling that out. As I left a little note for you there. And then what you have to do is build a taxon of character matrix. So you take all your characteristics and you put them across the top here, like I've done before you started to, teeth and spots. The outgroup, again, should have 0s across the board here, hopefully. And then you have your other groups, A, B, C, D, E, F, whatever. All right? But what you want to do is you want to first take this and probably draw it out on a scratch piece of paper. Why? Because you want to have the outgroup. Then you want the next one, whatever letter that is, maybe it's F, let's say that has the next least amount of characteristics, so or value. So as you look through here, let's say F had only spots. Okay, so everything else was 0 and it had 1. So this would be 0, 1, and let's say the next guy, he had 3 characteristics. He had 3, next one had 5, 7, 8, and 9. Okay, so you want your largest individual with the largest number of character traits at the bottom. This will help you build your phylogenetic tree at the bottom. And you can draw it on a piece of paper, I don't care how you do it, and take a picture of it and post it in there, or you can just build it in drawings, however you want to do it. Okay? So I'm sure there'll be some questions, but give it your best shot, and we'll see how it goes. All right? Is there any questions right now about the assignment that you can think of, or any questions about the CMU login or anything like that? If so, go ahead and ask. I will post the assignment for you here. This will be the last assignment that we have altogether this week. So just work on it. And like I said, we'll try to go over it a little bit on Thursday, look at the answer key, and go from there. I don't see any questions, but if you do have some, like I said, just email me, and definitely check out the CMU website for the course survey. Thank you guys for tuning in. We'll see you Thursday.